What if the most fundamental assumption that human beings have ever made about themselves turned out to be wrong? Not wrong in a minor, technical, footnote to a paper kind of way, wrong in the most profound, civilization reshaping, everything you thought you knew kind of way. What if the categories that humanity has used for centuries to divide itself, the races, the groups, the bloodlines, the deep biological distinctions that people have gone to war over, built empires around, justified slavery with, and organized entire societies upon? What if those categories, when placed under the most powerful scientific instruments ever pointed at human DNA, simply did not hold up? What if the genome, read in full, told a story so different from the one that human history has been telling that researchers had to sit in silence for a moment before they could articulate what they were seeing? That is exactly what happened. And the results were not subtle. They were not ambiguous. They were not the kind of findings that get lost in the supplementary materials of a specialist journal. They were findings so clear, so consistent, and so fundamentally at odds with how human beings have understood themselves that they have been described by the scientists who produced them as among the most important discoveries in the history of our species. The comparison of the complete genomes of populations from every major human group on Earth did not reveal the deep biological divisions that centuries of racial thinking assumed were there. What it revealed instead was something that the data makes impossible to argue with, something that changes everything. To understand why the results were so shocking, you first need to understand what scientists expected to find and what the long history of racial classification had led the world to believe was true. For most of recorded history, human beings have been categorized into distinct racial groups based on visible physical characteristics, primarily skin color, facial features, hair texture, and body proportions. These categories went by different names in different eras and different cultures, but the underlying assumption was always the same, that these visible differences reflected deep biological realities, that the groups were fundamentally distinct, that the boundaries between them were meaningful at the level of DNA itself, that what you could see on the outside was a reliable indicator of what was happening on the inside, at the most basic level of biological information. This assumption was not merely a social prejudice, though it was certainly that too. It was treated, for much of the modern scientific era, as an empirical claim. Racial scientists of the 18th and 19th centuries produced elaborate taxonomies of human types, assigning them ranks and capabilities based on physical measurements, skull shapes, and a cascade of pseudoscientific reasoning that we now recognize as one of the most catastrophic failures of intellectual history. But even after the explicit racism of those early taxonomists was discredited, the underlying intuition that the major racial groups represent genuinely distinct biological populations persisted in subtle forms throughout the 20th century. It shaped medical research, it influenced social policy, and it quietly underpinned enormous amounts of scientific work that assumed the racial categories were biologically real in ways that the genetics would eventually reveal they were not. The first major blow came with the work of geneticist Richard Lewontin in 1972. Lewontin analyzed genetic variation at a set of protein markers and found something that stunned the scientific community at the time. Approximately 85 to 90% of all human genetic variation exists within the populations traditionally called races, not between them. In other words, two people from what was conventionally classified as the same race could be, and routinely were, more genetically different from each other than either of them was from someone classified as belonging to a completely different race. The differences between the so-called major racial groups accounted for only 10 to 15% of total human genetic variation. The variation within those groups was vastly larger. This was not what anyone had expected, and it was only the beginning. As genetic technology advanced through the 1990s and 2000s and into the era of whole genome sequencing, the picture became not only clearer but more dramatic. The Human Genome Project, completed in draft form in 2001 and refined over the following decade, gave scientists access to the complete 3 billion letter code of the human genome. For the first time, it was possible not just to compare a handful of genetic markers between populations, but to compare entire genomes, all 3 billion base pairs, across individuals from populations spanning every corner of the Earth. The scale of the data was unprecedented. The computational power required to analyze it had only recently become available. And when it was applied to the question of racial biological distinction, the results were unambiguous. The total genetic difference between any two human beings anywhere on Earth, regardless of what racial category they are assigned to, is approximately 0.1%, 1/10 of 1%. The remaining 99.9% of the genome is identical across all human beings. When you compare that figure to other species, chimpanzees, our closest living relatives, differ from each other by about 1.2%. The implication is staggering. Human beings are one of the most genetically uniform large species on the planet. The diversity of visible appearance that human populations display, the range of skin colors from the deepest brown to the palest pink, the spectrum of facial structures, the variation in hair and eye color, the range of body proportions. All of that extraordinary visual variety is generated by a vanishingly small fraction of the genome. The overwhelming majority of human DNA is shared across every person alive. But the story does not stop at that 99.9%. When scientists began mapping the variation that does exist within that 0.1%, they found a pattern that completely dismantled the racial framework. If the traditional racial categories, broadly African, European, East Asian, South Asian, Native American, and so on, were genuine deep biological divisions, you would expect the genetic variation within each group to be relatively low and the variation between groups to be relatively high. You would expect clean boundaries. You would expect the populations of Africa to be more similar to each other than to the populations of Europe. You would expect the populations of East Asia to cluster together tightly, distinct from the rest. What actually happens when you plot the full genomic data is almost the opposite. African populations, rather than forming a single genetic cluster, contain the vast majority of human genetic diversity. The genetic distance between two individuals from different African ethnic groups, say a Yoruba person from Nigeria and a San person from Southern Africa, is frequently greater than the genetic distance between a European and an East Asian. This is because modern humans originated in Africa and lived there for the vast majority of our evolutionary history. All of the non-African populations of the world are descended from a relatively small group of people who left Africa between 50,000 and 70,000 years ago. That founding group carried only a subset of the genetic diversity that existed in Africa. As a result, every non-African population on Earth, every European, every East Asian, every Native American, every person from any ancestry outside the African continent is, in genetic terms, a subset of African diversity, not a different branch of the human family, a branch that grew from one part of an African root. This finding has a name in population genetics. It is called the Out of Africa model, and it is now supported by such an overwhelming convergence of genetic, archaeological, and paleontological evidence that it is one of the most thoroughly confirmed hypotheses in all of science. And its implications for the concept of race are devastating. If all non-African populations are genetic subsets of African diversity, then the category of African as a racial group is not a meaningful biological unit in the way that racial thinking assumed. It is the most genetically diverse major human grouping on Earth, containing within it more human genetic variation than all other regions combined. Any racial taxonomy that treats Africans as one category while treating Europeans or Asians as others has a proportionality exactly backwards. The situation is further complicated and further illuminated by what the ancient DNA revolution has revealed about the deep history of the so-called racial groups. Over the past 15 years, scientists have extracted and analyzed the genomes of ancient humans from archaeological sites across the world, giving us direct access to the genetic composition of populations as they existed thousands and tens of thousands of years ago. What this ancient DNA has revealed is that every major modern population is itself the product of multiple earlier populations mixing together. Often populations that were more genetically distinct from each other than the modern groups that resulted from their mixing are from their geographic neighbors. The populations that ancient racial science treated as primordial, original, and distinct were themselves the products of ancient mixtures. The categories that racial classification assumed were fixed and ancient were in fact recent and fluid. The people of modern Europe, for example, are not the descendants of a single ancient European population. They are the genetic product of at least three major ancestry streams: ancient Western European hunter-gatherers, early Anatolian farmers who migrated into Europe from the Middle East around 9,000 years ago, and steppe pastoralists from the Eurasian grasslands who arrived around 5,000 years ago. These three populations were more genetically different from each other at the time of their mixing than modern Europeans are from modern Middle Easterners or East Asians. Europe as a genetic region did not exist in its current form until these three populations mixed and their descendants became the dominant population of the continent. The European race, in other words, is a historical construction of the last few thousand years, not an ancient biological reality. The same pattern holds everywhere that ancient DNA has been studied. The populations of South Asia are the product of at least three major ancient mixing events. The populations of East Asia show complex layering of ancient genetic streams that defies simple classification. The populations of the Americas are descended from multiple ancient Northeast Asian lineages that are only now being fully disentangled. Every major modern human population, without exception, is a palimpsest of ancient mixtures, layer upon layer of different peoples coming together, mixing, diverging, and mixing again across timescales that dwarf the entire span of recorded history. This is the context in which the results of comparing every major human population's genome must be understood. The data does not show a set of clean, distinct biological groups with clear boundaries between them. It shows a continuous spectrum of genetic variation that reflects the history of human movement, mixture, and separation over hundreds of thousands of years. The boundaries that racial classification draws across this spectrum are not discoveries about nature. They are decisions about where to draw lines in a continuum that has no natural lines. Different researchers using different methods and different criteria produce different groupings, and all of them are partially right and partially arbitrary, because the underlying reality is not a set of discrete categories, but an endlessly complex gradient. There is one result from the full genome comparison studies that deserves particular attention, because it is perhaps the most personally significant for anyone trying to understand their own ancestry. When individuals from populations that are conventionally considered completely separate races are tested using the most comprehensive modern genomic tools, the level of ancestry sharing across conventional racial lines is far higher than most people expect. Studies of large population databases consistently find that a substantial proportion of people who identify as belonging to one racial group carry measurable genetic ancestry from populations that racial classification places in entirely different categories. The populations of the world have been mixing, trading, migrating, and intermarrying for so long and across such vast distances that the clean racial genealogies that many people believe they carry are in almost every case a significant oversimplification of a much more complex and much more interconnected genetic reality. The scientific consensus that has emerged from all of this work is now clear and consistent across research teams, institutions, and countries. Race, as a biological category, does not correspond to any meaningful division in human genetic data. The groupings that exist in the data reflect geography and history, the patterns of human migration and mixing over time, but they do not form the discrete, bounded, hierarchically organized system that racial thinking assumes. There are no genetic variants that uniquely define any racial group. There are no biological boundaries between the conventional racial categories. There are no pure racial lineages, no original racial types, and no genetic basis for any of the hierarchical rankings that racial science historically attempted to construct. None of this means that human genetic variation does not exist or is not real. It does exist. It is real. The patterns of variation that exist in the human genome are informative about ancestry, about the history of populations, and about certain medically relevant traits. But those patterns are not the patterns that racial classification predicted or assumed. They are more continuous, more geographically gradual, more historically complex, and more deeply interconnected than any racial framework has ever been able to capture. And the most important finding of all, the one that is most impossible to argue with once the data is in front of you, is that the genetic differences between the conventionally defined racial groups are among the smallest and least significant patterns in the entire data set. The thing that racial thinking placed at the center of human biological difference turns out to occupy the margins of human genetic reality. What does shock the people who encounter this data for the first time including many scientists who come to genomics from fields where racial categories are still used unreflectively, is not that human beings are genetically different. It is how genetically similar they are. The faces that look so different, the skin tones that span such a visible spectrum, the histories that have been so violently divided, all of them are expressions of a genetic difference so small that it would barely register against the backdrop of almost any other species on Earth. We are, at the level of the genome, one people, one recent, tightly related, extraordinarily interconnected family. And the sooner the world understands what the DNA has been telling us for decades, the sooner we might begin to build a civilization that reflects what we actually are, rather than the division we imagined. The DNA of every race on Earth has been compared. The results are in. And what they say, quietly, precisely, in the language of base pairs and allele frequencies and population structure analyses, is that the race was never real. Only the people were. But to leave the story, there would be to leave out the part that is perhaps most immediately relevant to the world we live in today. Because the scientific demolition of biological race does not mean that race has no consequences. It does not mean that the lived experiences of people who have been classified, judged, persecuted, and privileged on the basis of racial categories are somehow invalidated by the genomic data. It means something more specific and more important, that those consequences, the violence, the inequality, the hierarchy, the exclusion, were never the product of biology. They were always the product of choice, of power, of the decision by some groups to impose a classification system on others and enforce it with every tool available, from law to language to violence, for centuries. The genome proves that there was never any natural foundation beneath that system. It was always entirely constructed, and constructed things can be deconstructed. The medical implications of these findings are also profound and have been reshaping clinical practice for years. For most of the 20th century, medical research routinely used racial categories as biological proxies, assuming that differences in disease rates, drug responses, and health outcomes between racial groups reflected genuine biological differences between the groups rather than the effects of social inequality, differential access to health care, environmental exposure to stress and pollution, and the accumulated physiological effects of discrimination. The genomic data has forced a fundamental reassessment of this assumption. When researchers control properly for actual genetic ancestry, using real genomic data rather than self-reported racial identity, many of the health differences that appeared to be racial turn out to be the product of environmental and social factors that have nothing to do with DNA. And in cases where genuine genetic variants do contribute to different disease risks in different populations, those variants do not follow racial lines. They follow the actual patterns of population history and migration, which are far more complex than any racial taxonomy can capture. This is not a hypothetical or theoretical problem. It has had real clinical consequences. Medical guidelines that use racial categories as shortcuts for genetic information produced recommendations that were less accurate than guidelines based on actual genetic data, and in some cases, contributed to worse outcomes for patients. The ongoing project of replacing racial proxies with genuine genomic data in medical practice is one of the most important applications of the population genetics revolution, and one of the clearest demonstrations that the scientific dismantling of biological race is not merely an academic exercise, but a project with direct consequences for human health and well-being. There is also the question of what the genetic data tells us about human cognitive and behavioral variation, the domain in which racial pseudoscience has historically done the most damage, through claims about differential intelligence, temperament, and capacity between racial groups. The answer from the genome is unambiguous. The genetic variants that influence complex cognitive traits like general intelligence are distributed across the entire human population, without any pattern that corresponds to racial classification. There is no version of the genomic data in which any major population group carries a higher frequency of intelligence-associated variants than any other. The cognitive differences that exist between individuals are real and substantially heritable, but they exist between individuals, not between racial groups, and the genomic data provides no support whatsoever for any claim of genetically based cognitive differences between populations. The final dimension of this story that demands attention is perhaps the most ancient. The reason human beings are so genetically similar, The reason the 0.1% of genomic difference spans the entire species, with so little of it allocated to the divisions that racial thinking considered so fundamental, is that we are a young species. Homo sapiens, as we are defined anatomically and genetically, emerged in Africa only about 300,000 years ago. The populations that eventually spread across the entire planet descended from groups that left Africa within the last 70,000 years, an eye blink in evolutionary time. We have not had time to accumulate the kind of deep genetic divergence that would make the racial categories biologically meaningful. The chimpanzees who diverged from our shared ancestor 6 million years ago show more genetic difference between their populations than we do between ours. We are too new, too recently connected, and too persistently mobile and mixing as a species to have developed real biological races in the way that the concept implies. This is, in the end, the most astonishing thing the DNA comparison reveals. Not that we are all different, not that our genetic histories are complex and layered and fascinating, but that we are, for all the apparent diversity of our appearances and the genuine complexity of our histories, essentially one thing. One recent, remarkably uniform, relentlessly interconnected family of primates who spread across an entire planet in the geological equivalent of a moment and then spent most of our subsequent history convinced that the slight variations we developed along the way were fundamental divisions in our nature. The genome says otherwise. The genome says we are overwhelmingly the same. And the sooner we take that seriously, really seriously, not just as a talking point, but as the organizing principle of how we build our institutions and tell our stories, the closer we will be to living in a world that matches the reality that science has been describing for decades. The results shocked everyone. They should not have. The truth was always in our DNA. We just had to develop the tools to read it.